Hi everyone, in this video I am going to explain you about stem anatomy. Under this I will explain you transfer section of dicot stem and transfer section of monocot stem.
Internal structure of typical dicot stem. The internal structure of a typical dicot stem shows following features. The very first one, epidermis. Epidermis is the outermost layer of the stem. It is uniseriate, which means single layered. The epidermis of dicotyledonous stem shows multicellular hair-like outgrowths which are called trichomes. And the stomata also found in epidermis of dicot stem. The outer side of epidermis, a layer is present which is made up of the substance cutin. This layer is called cuticle. Epidermis plays a significant role in the protection of dicot stem. Remember this. Epidermis of dicot stem is uniseriate or single layered, but it contains multicellular hair-like structures called trichomes. Stomata also found in epidermis of dicot stem, and one more important thing is the epidermis of dicot stem covered by cuticle. The function of epidermis is protection. The second one is cortex. In dicot stem, cortex is divided into three important parts such as hypodermis, general cortex and endodermis. Hypodermis. It is present just below to the epidermis. The hypodermis of dicot stem provides mechanical support to young stem. This layer composed of collenchyma and their cells contains chloroplast. So hypodermis is green and photosynthetic. Cortex differentiated into hypodermis, general cortex, endodermis. Hypodermis present just below to the epidermis and it is composed with collenchyma. It gives mechanical support to young stems. Hypodermis contains chloroplast. Due to that, it is green and photosynthetic and prepares food material by wound. Next one, general cortex. This part is composed of parenchyma. Storage of food is the main function of cortex. Resin canal or mucilage canal present in general cortex. General cortex is schizogenous in origin. General cortex is composed of parenchyma. Storage of food is the main function of this region. Resin canal or mucilage canal also present in general cortex. General cortex schizogenous in origin. Next one, endodermis. Endodermis is single layer, but it is thick in nature. The cells of endodermis are barrel shaped. These cells accumulate starch in stem of dicot. Thus, this endodermis is known as starch sheath. Endodermis is innermost layer of cortex. It is single layered and contains thick walled cells. Endodermis cells are barrel shaped and these cells accumulate starch in stem of dicot. Due to this, endodermis is called starch sheath in dicot stem. Next one is pericycle. Pericycle is present on the inner side of the endodermis and above the phloem in the form of semi-lunar patches of sclerenchyma. In dicotyledonous stem, pericycle appears like semi-lunar patches present inside to endodermis and outside or above the phloem. And the important thing is the pericycle of dicot stem mostly made up of sclerenchyma. Note, in sunflower stem, pericycle is made up of alternate bands of parenchymus and sclerenchymus cells, in which pericycle which is present in front of vascular bundle is made up of sclerenchyma and remaining part is composed of parenchyma. Pericycle is heterogeneous in origin in sunflower stem. Next one, vascular bundles. Large number of vascular bundles are arranged in ring-like structure in dicot stem, which is called U-stele. The shape of vascular bundles are wedge-shaped. Each vascular bundle is conjoint, collateral, and open. Conjoint in the sense both xylem and phloem situated or lied in same radius. Collateral in the sense vascular bundle contains xylem and phloem are in 1:1 ratio. Xylem present towards center, phloem present towards periphery. And open vascular bundles. The vascular bundles which contains cambium between xylem and phloem are called open vascular bundles. Open vascular bundle shows secondary growth. Each vascular bundle is made up of phloem, cambium and xylem. Eustyl is present in dicotyledonous stem. Remember, the dicotyledonous stem vascular bundles are wedge-shaped, arranged in ring-like structure called eustyl. These vascular bundles are conjoint, collateral and open vascular bundles. The next one is pith or medulla. This is well-developed region or spreading from ring of vascular bundle to the center in dicot stem. The cells of this region mainly made up of parenchyma. The function of pith or medulla is storage of water and food. Note: the part of pith or medulla which is radially arranged between the vascular bundles are called medullary rays or pith rays. The main function of medullary rays is radial conduction of food and water. Here you have to remember the pith or medulla is well developed in dicot stem and it is made up of parenchyma. The function of medulla is storage of water and food. The parenchymal cells which are radially arranged between vascular bundles is called pith rays or medullary rays. The major function of these medullary rays is radial conduction of water and food. Let's see the diagrammatic representation of transverse section of dicot stem and ground plan or outline structure of transverse section of dicot stem. See, the outermost layer is epidermis. Epidermis covered by a layer which is called cuticle, and dicot stem shows multicellular hair-like outgrowths called trichomes. Below to epidermis, colonchymous layers are there which is called hypodermis. Then below to that it is general cortex made up of parenchyma cells. Resin canals or mucilage canals are present. Below to that single layer which is innermost layer of cortex is called endodermis. Endodermis cells accumulate with starch, that's why it is called starch sheath. Inner to endodermis is pericycle which is semilunar patches of sclerenchyma, then phloem, then xylem. Cambium is present between xylem and phloem. Here protoxylem present towards center, metaxylem present towards periphery. Then pith or medulla. See in ground plan or outline of dicot stem, the parenchyma cells between vascular bundles are called pith rays or medullary rays. Let's check this diagram carefully for your better understanding. Internal structure of monocot stem. The internal structure of a typical monocot stem shows following features. Under this, the very first one is epidermis. Epidermis is the outermost layer of monocot stem. It is uniseriate or single layer. Multicellular hair which are called trichomes are absent. Stomata also very less in number or may be absent. Outside of epidermis, a layer is present which is made up of cutin is called cuticle. Epidermis plays a significant role in protection of stem. Remember, in monocot stem, epidermis is as usual single layer, outermost layer, but trichomes are absent. Stomata is less in number or absent. Cuticle is present. The main role or the main function of the monocotyledonous stem epidermis is protection. Second one, hypodermis. In monocot stem, hypodermis is 2 to 3 layered. and just present below to the epidermis. The hypodermis of monocot stem is completely composed with sclerenchyma cells, and the main function of this hypodermis is providing mechanical support to the stem. Third one, ground tissue. The entire mass of parenchyma cells next to hypodermis and extending to the center is called ground tissue. There is no differentiation of ground tissue in monocot stem, but peripheral side cells are smaller ones and central side cells are bigger in size. Here you have to remember, hypodermis of monocot stem is completely composed of sclerenchyma, and function of this hypodermis is giving mechanical support to the stem. Coming to ground tissue, it is completely composed of parenchyma cells. These are present up to the center. And one more thing, the parenchyma cells of ground tissue which are situated peripheral sides are smaller ones, which are situated central side, those are bigger in size. Fourth one, vascular bundles. Many vascular bundles are present in monocot stem. Each vascular bundle surrounded by a sclerenchymaceous bundle sheath. All these vascular bundles are arranged in scattered manner in ground tissue. That's why this type of vascular bundles are still called atactostele. In monocot stem, vascular bundles are generally oval in shape. Each vascular bundle is conjoined, which means xylem and phloem both are arranged in same radius. Collateral, xylem and phloem are in 1:1 ratio, and closed, which means there is no differentiation, there is no cambium between xylem and phloem. Closed vascular bundles does not show secondary growth. Here we have to remember, peripheral vascular bundles are generally smaller than centrally located vascular bundles, which means peripheral vascular bundles are small in size, central vascular bundles are bigger in size. Remember, vascular bundles in monocotyledonous are large in number. These vascular bundles are surrounded by sclerenchymus bundle sheath. All these vascular bundles are arranged in scattered manner in ground tissue, which is called atactostele. The peripheral side vascular bundles are small in size, and centrally located vascular bundles are big in size. All the vascular bundles are oval in shape. Each vascular bundle is conjoined, collateral and closed type of vascular bundles. Let's see the diagrammatic representation of vascular bundle, which is in round or oval shape. So these vascular bundle covered by a sclerenchymus protecting sheath, which is called bundle sheath. Within that, they contains conjoined, collateral, closed type of vascular bundles. Here xylem and phloem both are present in same radius, but there is no cambium between xylem and phloem. That's why these are called closed type of vascular bundles, which does not shows secondary growth. Under vascular bundles, we have to know xylem and phloem. Xylem. In xylem, number of vessels are very less. In metaxylem, there are two large xylem vessels are present, while in protoxylem, there are only one or two but small xylem vessels are present. Xylem vessels are arranged in V or Y shape in vascular bundles. Just beneath protoxylem vessels, there are a water cavity which is cyzo-lysogenous in origin, but major part of water cavity is lysogenous in origin. This cavity is formed by disintegration of the element present below the protoxylem and neighboring parenchyma cells. Exception: there is no water cavity and bundle sheath in the plant called asparagus. The next one is phloem. Phloem consists of sieve tube elements and companion cells, but phloem parenchyma is absent. The next part is stele. Attactor stele is found in monocotyledonous plant. Attactor stele in the sense the scattered arrangement of vascular bundles in ground tissue of monocot stem is called attactor stele. Attactor stele is the most developed stele in plants. Let's see the diagrammatic representation of transverse section of monocot stem. See, in transverse section diagram, the outermost layer is epidermis. On the epidermis, cuticle is present. Below to the epidermis, there are 2 to 3 layers of sclerenchymus cells which are called hypodermis, which gives mechanical support to the stem. Below to that, parenchymus cells are there which are called ground tissue. In the ground tissue, several vascular bundles are arranged in scattered manner, which is called attactor stele. So vascular bundles are conjoined, collateral, closed type. See, the next diagram is outline or ground plan of transverse section of monocot stem. For your better understanding, let's check these diagrams carefully.